Good day, my dear students. I wish you happy holiday. So long. We are meeting again this moment for BCH 407. This is lecture 8 of the series of biochemical pharmacology, and the topic is inhibitors and physiological functions of cytochrome P450. This lecture will be quite short but meaningful. At the end, I expect that you get the best out of it. By way of definition, hemoprotein superfamily found across all domains of life are referred to as cytochrome P450, named for absorbing light at 450 nanometers when bound to carbon monoxide or carbon 2 oxide. The function in the synthesis of cholesterol, steroids, and lipid-derived signal molecules. They are also involved in detoxification reactions of drugs and other environmental toxins. Now, let's look at the objective of these lectures. Upon completion of this lecture, you should be able to outline the major stages and key enzymes involved in the de novo synthesis of cholesterol from acetyl-CoA. Describe the structure of cholesterol and its molecule as a precursor for steroid hormones and other important molecules. Explain the general pathway for the synthesis of the 5 major classes of steroid hormones from cholesterol. In addition, you should be able to identify the primary sites of synthesis for each of the major classes of steroid hormones. 5. Discuss the role of cholesterol, steroid hormones, and eicosanoids in various physiological functions or processes. We conclude by looking at how cholesterol synthesis is regulated. Starting right away with biosynthesis of cholesterol. The raw material is acetyl-CoA and acetoacetyl-CoA. Now, that gives condensation of that molecule with HMG-CoA synthase will give us 3-hydroxyl 3-methyl glutaryl CoA or HMG-CoA. That is further acted upon by HMG-CoA reductase to give us mevalonic acid. The next step is energy requiring in the form of ATP and is catalyzed by mevalonate kinase to give us mevalonate 5-phosphate. The next stage is catalyzed by phosphomevalonate kinase to give us mevalonate 5-pyrophosphate. That molecule will lose carbon 4 oxide by the action of mevalonate 5-pyrophosphate decarboxylase to give us isopentenyl 5-pyrophosphate PP. Now, that molecule is acted upon by farnesyl PP synthase to give us geranyl pyrophosphate, and the same enzyme, farnesyl pyrophosphate synthase will act on it to give us farnesyl pyrophosphate. From there, squalene synthase will form squalene. Then squalene monophosphate monooxygenase will give us 2,3 dioxidosqualene. Now that one will be oxidized to give us squalene epoxidase. Now the action of squalene epoxidase will give us lanosterol. Now that lanosterol will further undergo 19 reactions. You can see how complex the synthesis of cholesterol is. It will undergo 19 further reactions before it will get to cholesterol. Now, what is this cholesterol and what shape does it have? What is the structure of cholesterol? This is the structural backbone of cholesterol. The one that is painted in pink. That is the backbone of cholesterol. It has a polar head and a non-polar body. It is this amphipathic nature that is crucial for its role in cell membrane, where it helps to modulate membrane fluidity and stability. Of course, cholesterol structure has two main components or domains. First is the steroid nucleus, which is shown in pink, and the second is the hydrocarbon side chain. It is this hydrocarbon side chain that is the hydrophobic aspect or region of the molecule. In vertebrates, cholesterol is the central precursor of all steroids. It is secreted by the testes of the male, the ovaries of the female, and the adrenal of both sexes. These tissues share many enzymes for the transformation of cholesterol. A major common pathway involves conversion of cholesterol to progesterone. Important molecules derived from cholesterol. There are other important molecules that are derived from cholesterol. Examples are the bile acid, vitamin D, and others like the eicosanoid, the inflammatory molecules. Now, the liver synthesizes cholic acid and chenodeoxycholic acid from cholesterol. Vitamin D3 is synthesized in the skin from 7-dehydrocholesterol, a cholesterol derivative. And cholesterol itself is a key structural component of all animal cells, where it maintains fluidity, permeability, and cell integrity. Let's look at the synthesis of steroid hormones. Like I said earlier, cholesterol is the backbone of all steroids. The 5 major classes include glucocorticoids, that is exemplified by cortisol, which regulate metabolism, inflammation, and immune response. Mineralocorticoids, exemplified also by aldosterone, controls electrolytes and water balance. Androgens, example is testosterone, mediates the male sex hormones, which is involved in reproduction, and secondary sexual characteristics. Estrogen, exemplified by estradiol, which is the female sex hormones regulating reproductive functions, and progestogens, exemplified again by progesterone, which maintains pregnancy and menstrual cycle. Now, talking about the biosynthesis or biosynthetic pathway, cholesterol is the backbone or the starting material. It gives, in the process, gives pregnenolone, progesterone, and aldosterone and cortisol. Now, this is the pathway. Now, starting from cholesterol, you can actually have two pathways from pregnenolone, one leading to aldosterone and the other leading to cortisol. And the enzyme catalyzing each reaction is stated. You take your time to study this pathway and take a good note of it. However, it must be said that these enzymes are all part of the cytochrome P450 enzymes. For the synthesis of testosterone and estradiol, the pathway progresses from cholesterol to pregnenolone, from pregnenolone to testosterone, then from there to 7-hydroxyprogesterone to androstenedione. From androstenedione, it is now converted to testosterone, and from testosterone to estradiol. The enzymes catalyzing each reacting step is indicated, and their cytochrome P450 isoenzyme indicated. Let's take time to explain this pathway. I've consistently said that cholesterol, for the synthesis of these steroid hormones, cholesterol is the common starting molecule. The process is regulated by ACTH, which stimulates CAMP to activate the cytochrome P450 side chain cleavage, that is P450-SCC, cytochrome P450 side chain cleavage, and that is the rate limiting step in the reaction, or the rate limiting enzyme in the reaction. The primary site of synthesis include adrenal cortex for aldosterone and cortisol, then the gonads for androgens and estrogens. So the locations of this synthesis or synthetic site is now known. Now, let's look at the mineralocorticoids pathway exemplified by aldosterone production. It is not different from what I have been saying. Look at the pathway, follow the trend, you will see that this has been said before now. I just highlighted the pathway to distinct this mineralocorticoid pathway synthesis or mineralocorticoid synthetic pathway. And mineralocorticoids are involved in regulation of salt or sodium and blood pressure. The same happens with the glucocorticoid pathway, that is the pathway that um, leads to the production of cortisol. Now, cortisol functions to manage stress response, metabolism, and immune regulation. Androgen and estrogen pathway, that is the pathway leading from testosterone to estradiol. This is a pathway, we have seen it before, it's on the other um, slide. This is just by way of bringing out the specific pathway for your notification and learning. Now, what could be the role of cholesterol and some of its derived lipids? Cholesterol, steroid hormones, and eicosanoids are all lipids that play a crucial role in various physiological processes. For example, cholesterol is involved in cell membrane stability, precursor to steroid hormones, bile acid, and vitamin D, among others. Steroid hormones, which is also derived from cholesterol, is involved in the regulation, production, stress, and electrolyte balance. While eicosanoids are lipids or cholesterol-derived molecules, lipids, that are involved in inflammation, immunity, blood clotting, smooth muscle functions. You can see how important this molecule called cholesterol is. How is the synthesis of these all-powerful cholesterol synthesis regulated? The synthesis is highly regulated and primarily controlled at the level of the enzyme MGH-CoA reductase, which catalyzes the conversion of MHMG-CoA to mevalonate. I mentioned earlier that that stage or that step is the rate limiting step in cholesterol biosynthesis. When we say rate limiting step, it is irreversible and is always by feedback inhibition. These are other types of control or regulation tabulated for your convenience. Now let's look at inhibitors of cytochrome P450. The isoforms are indicated here. Common inhibitors are also provided. For example, for cytochrome 1A2, these are molecules that can inhibit these enzymes. We refer to them simply as antibiotics. We have 1, 2, 3, 4, 5, 6. So you can, for example, cytochrome P450 2C19 inhibits the proton pump, is a proton pump inhibitor and an antidepressant. With that, my dear students, let us take a step towards self-assessment of our understanding of what has been provided. I have presented here 8 questions from these slides for which you can use to assess your understanding of the topic presented. I hope you will find it useful. In case of any question or misunderstanding which you need clarification from, having consulted with your colleagues or your fellow students, and you are not satisfied with the answers provided, you can do as usual, contact me through your class coordinator. You can also write questions and post so that I answer it through the same medium for others to benefit from. Thank you very much and have a nice day.